Hello everyone. In this video, we are going to see about origin of biopotential and its propagation. This is Dr. Inbamalar, working as professor in the Department of Electronics and Communication Engineering, R.M.K. College of Engineering and Technology. First, let us see what are biopotentials. Biopotentials are electrical potentials that are created by the flow of ions in and out of the cells. The flow of these charged ions creates potential differences between the inside and outside of cells. These potential differences are called as biopotentials. These biopotentials can be measured with electrodes and electronic instrumentation that provide insight into the functioning of various biological systems. Now let us see about the origin of bioelectric potentials. Certain types of cells within the body, such as nerve and muscle cells, are encased in a semi-permeable membrane. This membrane permits some substances to pass through while others are kept out. Surrounding the cells of the body are the body fluids. These fluids are conductive solutions containing charged atoms known as ions. Bioelectric potentials are created by the flow of ions in and out of the cells. The flow of these charged ions creates potential differences between the inside and outside of the cells. These potential differences are called as biopotentials. As mentioned already, these can be measured and this will help the healthcare professionals to provide insight into the proper functioning of various biological systems. These biopotentials can be studied under two headings. The first one is resting potentials. The principal ions are sodium, potassium and chloride. The membrane of excitable cells permit entry of potassium and chloride ions, but it blocks the entry of sodium ions. So, the concentration of sodium ions inside the cell becomes much lower than that of the intercellular fluid outside. Since the sodium ions are positive, this would tend to make the outside of cell more positive than inside. Additional potassium ions, which are also positive, enter the cell, causing a higher concentration of potassium on the inside than on outside. This charge balance cannot be achieved. Equilibrium is reached with a potential difference across the membrane, negative on the inside and positive on the outside. This membrane potential is called resting potentials. This potential is measured from inside the cell with respect to the body fluids. So, the resting potential of a cell is always negative. This resting potential value ranges from minus 60 millivolt to minus 100 millivolt. The cell in the resting state is called polarized cell and this process is called polarization. This diagram shows the resting potential. Here, the inside of the cell is negative and the outside of the cell is positive. The electric potential is measured. It is on an average minus 70 millivolt. Now, let us see about action potentials. When a cell is excited, the membrane changes its characteristic. It begins to allow some of the sodium ions to enter. The sodium ions are rushed into the cell. At the same time, potassium ions move from inside but unable to move as rapidly as sodium ions. As a result, the cell has slightly positive potential inside due to the imbalance of potassium ions. This potential is known as action potential. It is approximately plus 20 millivolt. This diagram depicts the action potential. As you can see in the diagram, inside of the cell is positive and outside is negative. It measures around plus 20 millivolt. A cell that has been excited and that displays an action potential is said to be depolarized. Now let us see about repolarization. Once the rush of sodium ions through the cell membrane has stopped, the membrane reverts back to its virginal selectively permeable condition. Here, the passage of sodium ions from the outside to the inside of the cell is again blocked. However, it would take a long time for a resting potential to develop again, but such is not the case. By an active process called sodium pump, the sodium ions are quickly transported to the outside of the cell and the cell again becomes polarized and assumes its resting potential. This process is called repolarization. Cell comes from depolarized state into polarized state is called repolarization. This diagram depicts the sodium pump or otherwise called as sodium-potassium pump. Here, the sodium-potassium pump binds 3 sodium ions and a molecule of ATP. The splitting of ATP provides energy to change the shape of the channel. The sodium ions are driven through the channel. The sodium ions are released to the outside of the membrane and the new shape of the channel allows 2 potassium ions to bind. The release of the phosphate allows the channel to revert to its original form, releasing the potassium ions on the inside of the membrane. This diagram shows the resting and action potentials along with depolarization and repolarization. So from the graph it is understood initially when time is equal to 0, the cell is in the resting potential which measures approximately minus 70 millivolt. Then due to depolarization it reaches the action potential with an amplitude of plus 30 millivolt. Then repolarization brings it back to the resting potential condition. Now let us see some definitions related to this bioelectric potentials. First, let us see all or nothing law. Regardless of the method by which a cell is excited or the intensity of the stimulus, action potential is always the same for any given cell. This is known as all or nothing law. Net height of action potential. Net height of action potential is defined as the difference between the potential of the depolarized membrane at the peak of action potential and resting potential. Absolute refractory period. Following the generation of an action potential, there is a brief period of time during which the cell cannot respond to any new stimulus. This period is called absolute refractory period. Relative refractory period. Following absolute refractory period, there occurs a period during which another action potential can be triggered but a much stronger stimulation is required. This period is called relative refractory period. Now let us see some of the examples for biopotential signals. First one is action potential, which can be generated by nerve cells or muscle cells. Then electrocardiogram, whose source is heart. Electromyogram generated from the muscles. Electroencephalogram are the signals from the brain. Electro-optogram are the signals from the eye dipole field. Electro-retinogram are the bioelectric signals from the eye retina. Electrogastrogram are the signals from the stomach. And galvanic skin reflex are the signals from the skin. Thank you all for watching my video.